Okay, so welcome today. We have with us Dr. Will Rust. He is CEO of Seraxis, which is a company that's aimed at pancreatic islet replacement therapy. A little bit about Will. He is the co-founder and CEO of Seraxis, as I mentioned. After raising seed funds to launch Seraxis in 2013, he engineered a stem cell line with enhanced pancreatic differentiation potential as a basis for anti-diabetes islet replacement therapy. Seraxis subsequently completed 3 rounds of VC funding, developed cGMP for manufacturing of highly pure and potent replacement pancreatic islets, and is preparing for clinical testing this year. In parallel, Seraxis has developed 2 proprietary methods of immune evasion, and so we're really looking forward to hearing more about the company and the progress. And welcome, Will. Very nice to see you again. We've had you last year, earlier in the last year, so it'd be great to bring us up to speed.
Well, thank you, Monica. I appreciate the opportunity to be here and do that. So let me jump into my presentation. Like Monica said, we are focused on developing a cell therapy. It's a replacement islet cell therapy for type 1 diabetes. And what I'm going to do is show you the progress that we are making towards our first clinical study. So here we go. Here's our clinical goals. So the first clinical study that we will be launching, which we're going to file an IND for very soon, we're just a month or 2 away from that, is a replacement pancreatic islet that would be given to patients who will require immune suppression. So these are patients that suffer from severe recurrent hypoglycemia. And then we'll follow that up with a second program using virtually the same strategy, but with a cell therapy that has been genetically modified to evade immune detection. So this would be given to patients with a reduced or absent immune suppression. All right. So, all right, let me go into tell you how we do it, why we're different. So the strategy I think you're aware of, everybody knows that islet transplant therapy can be curative for patients that require insulin. In fact, this therapy using islets derived from cadaveric organ donors was approved last year by the FDA, and that product is called Lantidra. And the process is well known now. So once a donor pancreas was received, the islets are isolated and then transplanted to the patient in need through the portal circulation. So these are introduced into the blood supply that feeds the liver, and some of them survive that process. They get trapped in the microvasculature, and after that heals, they're actually inside the liver, and that converts the liver into an organ with 2 functions, obviously liver function, but also endocrine pancreas function, and those patients require immune suppression. So what we do at Seraxis is try to improve on this method, on this process. The first improvement is a replenishable supply of islets. So instead of getting them from organ donation and harvesting from those organs, we have a stem cell line, and we use that to grow replacement islets in a GMP facility to have clinic-ready replacement islets for as many people as needed. And the other things that we do to improve on this process is we don't deliver to the liver. I'll tell you why there's a better site. And we also have the strategies for avoiding the immune suppression. So firstly, what we do is differently from what you've more familiar with, which is growing islets from pluripotent stem cells, such as embryonic or iPS cells. Here's why what we do is different. We have our own cell line that we made in 2013, and we use that as a basis for our manufacturing. It is not an iPS cell. It is not an embryonic stem cell. And here's where it came from. We took islets that were harvested from cadaveric pancreases acquired through organ donation, and we reprogrammed those individual cells into a self-replicating stem cell state. And then we screened those. There was hundreds of them that we produced. We screened them, trying to find the one that was just naturally very good at maturing back into its origin, right? Back into becoming an endocrine committed pancreatic cell. And after that screening process, we identified one that was absolutely the best at doing this, and it's called SR1423. That's those numbers come after the designation for the clone it came from. And we took that cell line, we made master cell and working cell banks and qualified those for clinical use. And that's what we are using now as the foundation for all of our islet cell manufacturing. And the stem cell is, in fact, very different. So it is not pluripotent. That's the number one issue. If we do an unsupervised analysis of the genes expressed by each individual cell, what we see is the top graph here, where our cells are colored blue, and then a reference data set from embryonic and iPS cells are colored in peach and green. And we can see that there's little overlap. It turns out that when you look specifically at the genes that define pluripotency, those genes are lacking in our cells. So they absolutely fail the criteria for pluripotency. There's a couple of other interesting genes that they lack. One of them is expression of brachyury, which is the master transcription factor that specifies most of the tissues of the human body. So all mesodermal tissues. Because it lacks expression of that gene, it will not differentiate along that path. So it is very limited in its fate potential. And that's why it's very good at making endocrine pancreas.
So you mean, so that's sort of like a protective factor against tumorigenesis.
So the lack of transcription factors that define pluripotency is, yes, a protective factor against tumorigenesis. So it's great that you bring that up, because iPS and embryonic stem cells are defined by their ability to form teratomas. So these are tumors that occur after the cells are implanted into animal models. And the reason that's important for an embryonic or an iPS cell is because it proves that that cell has the ability to form every tissue of the embryo. Ours won't do that. A consequence is that it won't form tumors at all. So we put them into these animals and they differentiate and they sit there. It won't form teratomas, they won't form embryos or and they don't form progressively growing tumors. So it makes it much, much safer as a starting point for a clinical cell therapy product than a pluripotent stem cell. It's an important distinction. So not only do we make pure islets with a much more streamlined differentiation protocol, but we lack that safety risk that is present with other cell types. So it's not just that's a difference. There's another thing that I want to focus on for a few minutes that we published late last year, and that is how we define the replacement pancreatic islet. So what's most common in the literature is to define the pancreatic islet based on expression of a transcription factor called NKX6.1. And NKX6.1 is expressed in the mature islet, and it is a required transcription factor. However, when we took NKX6.1 expressing cells and put them in animal models, what happened over time is that they extinguished insulin expression and failed to rescue diabetes reproducibly in those models. And so we began to question the potency of the NKX6.1 expressing cell. What we found was that the cells that had persistent insulin expression, they were actually rich in two other transcription factors, MAFA and ISL1. Turns out that those are more specific transcription factors that define the endocrine pancreatic cell fate than NKX6.1. So we switched gears and we changed our protocol so that we would manufacture cells that were, instead of NKX6.1, they were rich in ISL1 and MAFA. And it turns out that these were much more potent at rescuing diabetes in our animal models. And then upon sacrifice, we were able to remove grafts that really look exactly like an endocrine pancreas. And I'll show you some of that. But let me first, compare and contrast to embryonic and iPS cells. Here's a recent publication, also came out late last year, where it defines the exact difference I'm talking about. So the problem with NKX6.1 is that not only is it present in the beta cell, but it's also important to specify a different type of endocrine cell in the gut called an enterochromaffin cell. And it turns out that the embryonic stem cells and iPS cells that are expressing NKX6.1, they actually produce a lot of these enterochromaffin cells in the differentiation cultures. And this is an off-target contaminating cell. Those same cells that express a lot of NKX6.1 have very low level expression of MAFA. So that just highlights the two different populations of cells. The ones that we're more familiar with that express NKX6.1, and the one that we make at Seraxis, which does not express NKX6.1 and instead expresses MAFA. If you look on the left, this is from the publication that I referenced, where the enterochromaffin population is labeled EC. So you can see two populations here. This is single cell sequencing data where they're labeled in yellow and then light purple. And you can see the relative proportion of the enterochromaffin identified cell versus the actual therapeutic beta cell. And you can see that the presence of the enterochromaffin cell is actually pretty big. That's going to decrease the potency of the cell therapy. So this is one, I think, very important differentiating factor between what we do at Seraxis and what others are doing. When we do complete the manufacturing, the clusters we make look a lot like native islets. Can't really tell the difference under a microscope. And the expression of the proportion of the hormone expressing cells is very similar to a native islet. So on the graph on the right, this bar chart, I'm showing the percent of each islet that is comprised of these hormone expressing cells, somatostatin, insulin, glucagon, ghrelin, and pancreatic polypeptide. And it's very similar to a native islet. So the native islet in this graph is light gray, and ours are red. So we really do recapitulate and mimic that native islet. It also mimics the native islet in terms of performance in the lab when we measure the secretion of insulin. So here on the y-axis is C-peptide, and on the x-axis is varying levels of exposure to glucose. And we can see that insulin jumps in its secretion when they are first exposed to high glucose, and then that tapers off and follows a trowel when it's in low glucose. Just for comparison, on the left is an image that comes from a publication where this performance is tracked in human islets under the similar conditions in vitro. And you can just see that those patterns of insulin secretion are very similar.
Will, can you go back one slide?
Yeah, this one?
No, yeah, but no, the one more. Yeah. So these are the Seraxis SRO2, right?
Yeah, that's a pretty picture of one of our clusters. We make billions at a time, so that's kind of unfair, but yeah.
Okay, yeah, it looks very nice. So you're saying that these cells that you guys are manufacturing are able to really put out the different hormones that, physiologically, are made by different cell types.
Yes.
So how is that happening? Because you were first talking about just sort of the one cell type, right? The beta cell type, and yet you've got like the serotonin producers, the glucagon producers. You've got cells that are behaving like the other constituents of the islet, alpha,
So
delta, epsilon, PP cells.
Exactly. So we're not making just beta cells. What we have is a cell population committed to become the endocrine pancreas. And it does make all of the components of that endocrine pancreas and a few other pancreatic cell types that we've identified. So it has not just the beta cell. So this is not a beta cell replacement therapy, but rather the whole islet replacement therapy. And for a lot of reasons, that's very important. Physiologically, of course, we need all those cell types to have appropriate glucose, control, and there has to be communication between those cells as well. In our experience, the actual structure of the islet with those cells distributed appropriately is also important for their function. So it matters. Form and function in the human body is really important. Those things are related.
Okay,
and,
so
we have all different cells.
It just made a little bit of a jump because we're talking about the insulin cells producing
Yeah.
cells. And
So I
then,
was talking about NKX 6.1 and insulin secreting cells, and that's maybe why we are focused too much on just the beta cell. But the
yeah,
population of cells defined by expression of MAFA and ISL, so not every cell in those populations express those two, but they're very rich in that expression pattern. That population of cells gives rise to the insulin secreting beta cell, the glucagon secreting alpha cell, and the rest.
okay. And then those cells do in fact form, this cluster. Um,
Yes, yes,
so
they
that
do.
they're not standalone. I noticed you said before there was no tumorigenicity of the individual beta cells, but then I guess when you're adding them together, they are clustering.
That's right. So we never grow them as a single cell suspension at any point. It's always as aggregates. They grow, they differentiate this way. That crosstalk among cells is important for the process.
Great, got it. Thanks for clarifying.
So the performance is good. We have a manufacturing facility at our laboratories here in Maryland where now we manufacture those in a fully GMP compliant manner to support our non-clinical studies and then very soon our clinical program. And we're very proud of that because it's actually very difficult to manufacture what is in essence a replacement human organ in the laboratory and do it in a way that would be acceptable for regulators to approve for clinical use. So we did that. We had a press release earlier this month or earlier in January that highlighted the fact that we had an external audit performed on our GMP manufacturing facility, and that audit demonstrated that we were in fact clinic ready. We passed every criteria for that audit. When we put these GMP compliant clusters into animals, we were able to rescue diabetes at different sites and doses. And this is some data from a IND enabling study that was performed at a CRO on our behalf. And in this case, we provided STZ-induced diabetic mice with our clusters. And then, as you can see from these graphs, whether we put them in the kidney capsule or a surrogate for our preferred implant site, the fat pad, we were able to rescue diabetes in those animals long term. So back to this concept, I explained how we solved the first problem of a replenishable supply of islets. What I wanted to do was talk about how we've improved on the implant method and tell you how we intend to avoid immune suppression. So this actually is a very important part of what we do. Just as important as making MAFA, very pure, very potent islets in the laboratory. And I'll tell you why. First, we don't think that the liver is the best site, for treating most patients. And one of the reasons is that there's not a great volume in there to implant a therapeutic dose. It's very difficult. There are physiological risks associated with implanting to the liver, but it's also can't be monitored. You can't see it from outside the body, what might be happening with the implant and the graft. And it obviously can't be biopsied or resected either without serious risk to the patient. So what we did instead was try to find an alternate site. There's only one site that is actually proven in human patients to be adequate for a site to house a replacement pancreas, and that's the omentum. So we did a lot of work trying to understand how you take lab-grown islets and pair that with the host omental tissue to essentially remodel into a chimeric organ that is comprised of both host and donor, but has everything that a functional pancreas needs, right? Something that has vasculature, has three-dimensional soft tissue with parenchyma that keeps the islet separate, yet able to communicate the way a real pancreas does. And so, after refining this process, we figured out how to do it. And here's some examples of how it works. So on the left is a picture of a pancreas that we've remodeled from rat omentum. And on the right is a pancreas, extra pancreatic pancreas that we remodeled using our islets and the non-human primate omentum. And when we section these and look at them, under the microscope, what we can find is tissue that does have all the components of a functional pancreas. So this is from the rat. This is from the rat experiment. So you can see on the left, it's stained for chromogranin A in green. That's a pan-specific marker of the endocrine pancreas, and insulin in magenta, and blood vessels. And you can see that the islets have retained those cluster shapes, and then they are separated by host tissue that includes blood vessels. And that tissue in between, I can confirm, is rat tissue, not human. You can see that clearly on the right, where we have a similar section stained for human mitochondria in green. So that marks all the human tissue. And then the blood vessels in red. And the you can see that the tissue in between the islets are rat tissue. They're not stained positive with the human mitochondrial marker. And in fact, if we do this counter stain with a rodent-specific marker, then it is positive. So this is a true chimeric organ made of the host and the donor tissue. If we look at a non-human primate, pancreas that we generated from that tissue, it's very similar. On the left is staining insulin in magenta and NKX 6.1 in green. And this shows that the NKX 6.1, after the graft has been remodeled and matured and is functional in that animal, it does in fact turn on expression of NKX 6.1. And then on the right, I'm just showing you distribution of the various hormone secreting cells in those islets. So you can see it's not just the insulin secreting beta cells, but we also have the glucagon and somatostatin secreting cells in this image. And then back to where we're going with all this, that data that I presented and a whole ton of extra safety data, we're putting into a package to submit to the FDA right now in the form of our IND to support a phase 1/2 clinical study that we hope to get launched very soon. So that is our strategy. I'm probably not going as fast as I needed to. We're starting to run out of time. So let me just spend 1 minute telling you what our next plan is, and that's to do exactly the same thing I just showed you, using the same cells and the same manufacturing process at the same implant site in our clinical study, except that we have modified that parent line so that it should be much more acceptable as a graft to an allogeneic patient, an allogeneic donor to a patient, so that this patient would not need immune suppression or a tolerable amount of immune suppression. So we've completed those modifications, and we have made our cell banks, and we have begun testing of these cells. And it really is remarkable, because when we put these in animals that have been humanized, so for example, a mouse whose bone marrow has been irradiated and then seeded with human hematopoietic stem cells, so they have a nearly full functioning human immune system. When we put in our gene-edited cells, they're not able to stimulate a T-cell mediated immune response against the graft, whereas the parent line in a wild-type animal will, of course. I mean, a parent line in that animal with a functioning immune system will. And that's what those images on the top are showing. And when we do in vitro studies as well, we can show that our modified line cannot stimulate a T-cell mediated immune response. It won't activate those T-cells. So this is our next clinical program to follow up very closely on the first clinical program. And I think I used up all my time, but I got through my slides. So thank you very much.
That's great.
Just a few minutes for questions.
Yeah,
right?
there's a few questions. Here's I have a couple, and here's some more. Here's a question here from Alexis Carmer.
Hi, Will. Thanks. That was really interesting. The first question I had, you showed a nice slide that showed inclusion of the vasculature in that new organ that had formed, implanted in the omentum. I'm just curious, do you know if there's nerve tissue forming in that islet as well?
So I have not stained for that, but that is a very good question, because pancreatic islets are innervated, and we should look to see if there are axons that are growing and making a connection there. We have not done that yet.
I've got a couple more. One for your SR03 follow-on product, the hypoimmune product. I'm wondering if you feel, I don't know what kind of interactions you've had with CBER, But if you feel the pathway is well defined as far as inclusion, exclusion criteria, when you're not using immunosuppression or using less immunosuppression?
So we do feel very strongly about that. It's a calculation of risk benefit, right, for each patient. And
Mhm.
so what we need to do is ensure that the safety profile is very good. And that's what we've done. So that's one reason why it's important to use a cell line that's not tumorigenic. That's one reason why it's important to deliver to a site that can be monitored. And then in addition to those things, we actually have a safety net built in. So we had cause expression of yet another marker on the surface that would enable us to target these cells if we needed to. So all those things together, we feel very strongly pushes the risk reward benefit towards a profile where most patients could be included in either a clinical study or actually receiving that product once we become commercially available.
Just one more. I was wondering, have your cells been evaluated independently at all? I just noticed this week there was a paper, Horikawa and all, I think out of Japan, where they evaluated like 15 HIPSC cell lines for differentiation efficiency. Have your cells undergone any independent evaluations?
Yeah, we have done that. So we are not from a university lab with the reputation to draw from that. We're just a group of dedicated scientists here in our lab in Germantown who are focused on this because we're committed and we feel that it can be done. So yeah, when we went out to raise money, which we needed a lot of, the people with those kinds of deep pockets, they wanted to test our cells independently. So one group asked that we send it to a JDRF sponsored lab, which we did. One group asked us to send it to their experts in their own lab, which we did. And then we sent them to a collaborator, another collaborator that tried to reproduce what we did in a lab in South Korea. So all three of those places reproduced what the data that I've shown you today. And all three of them came back and said, not only were they able to reproduce what we told them, but they had never seen the same thing from an embryonic or an iPS cell, which is our experience too. We secrete 10 times more insulin than those cells do, and they're pure.
Exciting. Thank you.
Thanks for that. There is a question in the chat. Let's see here. That is, how far away would you estimate that your clinical studies on children will be coming? Is there some kind of timeline when you think the treatment will be, hitting the market? That's a big question.
Yeah, I don't even want to answer this question because it's kind of depressing. So the clinical studies we endeavor doing are expensive, right, and difficult. Patient recruitment is long and slow, and the outputs we're measuring are long-term outputs, and that's frustrating for families who are desperate for getting a cure. I mean, every stakeholder right now in this field is expecting a curative islet replacement therapy soon because the data is too strong. Proof of concept is already there. Everybody knows this can be curative. So where is the therapy that's accessible? And I feel, the same frustration as you do. The problem is that to get to children, it's going to need to go through a long path of previous studies in adults demonstrating, safety and durability and efficacy. And, imagine the regulatory agency, they're going to protect the health of children above every other patient population. So this is going to have to wind its way through adult patients first before I think that's even considered.
Okay, thanks for that. I just wondered, what are your thoughts of sort of the monitoring aspect of your implants, particularly those that are, immune cloaked? Do you have a strategy in place? Have you thought about, Katie Digovich's approach with Minutia? She's got those nanosensors already built, and they are, ready to sort of monitor implanted, islets and beta cells, et cetera.
So I am familiar with that. We are confident in our approach to be able to monitor the implant and then the graft that is formed after that implant is successfully passed through its maturation and so forth, and remodeling process. The omental tissue is in the abdomen right next to the pancreas, and it's in a site that can be detected through regular methods of looking at the abdominal organs from outside the body. So we feel confident that we can do that. There are also markers that can be analyzed in the blood that would easily demonstrate if there A was a problem, and B follow the performance of the graft to make sure that it's working as it's supposed to. So we'll do both of those things, both the imaging from outside and monitoring of the blood chemistry. So we feel that that's going to be enough for what we're trying to achieve.
With the imaging, what kind of, are you talking about, like what kind of scans are you speaking about? 'Cause sometimes-
Echocardiography, just using a sonogram, just like an obstetrician would do to look at the developing fetus.
Okay, and so then that would be a cost that insurance will have to digest, right? Sort of
Oh,
the-
I'm just looking as far as getting a successful clinical trial right now.
Right, right.
That's on us. We'll pay for that. Right?
Okay,
You're looking later at whether or not monitoring would be required in a patient once we reach commercial. I don't have any conjecture about that. I
yeah,
would guess that the answer is no. They would not need any monitoring beyond taking an occasional blood glucose strip to determine if it was still functioning.
all right, well, that's great. And then how about, replacement features? It looks, I think I saw 3 months as one kind of, lifespan of some of these implants, but what sort of, can you speculate on what kind of timeline might be available for patients for this product?
You mean how long it would last?
Yeah.
So, we don't have any idea. So the only thing we can benchmark against is cadaveric islets, and cadaveric islet longevity is in a large part determined by the immune suppression, because those are toxic to the islets. So it used to be after the first studies that by 2 years, half the patients already became insulin dependent again. Now things have improved, and I think that's because the better immune suppression regimens using less toxic drugs and better tailored for each patient. Now, at 5 years, you can have very good outcomes in the number of patients that remain insulin independent. But ours are not cadaveric islets that are fragile and harvested from a donor and have already suffered a great deal of stress and damage before even implant. So we could anticipate it would be longer than that, but we have no evidence for that. It has to be determined in a human patient. The animal models are too different, and besides, they're so sickly they usually die by 6 months anyway. So we just can't know until we determine it empirically.
Mm-hmm. Okay, I mean, it's very interesting. We'll see. We've got a couple of horses in the race now. Seraxis, Vertex, and others. Oh, another question.
One more, Will. I just read that AstraZeneca is investing in a cell therapy manufacturing facility in Maryland, right near you. Just
Right,
curious
this morning's news.
if you can share what your plans are for commercial production of your product.
Finding a partner who will help us make all of those. So that's going to be the easiest path. We're committed to doing it no matter what. So we have an arrangement with a CRO for large scale manufacturing. Right now, in our current facility, we can support the clinical studies that we have planned, but we're ready to pull the trigger on large scale manufacturing at a CMO that would be able to make thousands of doses. So it's just we've just got to figure out the right path for us. We're trying to do that right now. I think the most important thing is getting the clinical study launched, which is already an exhausting endeavor. All of us are focused 100% on that goal right now.
Fantastic. Well, you've got a new facility in your backyard, so that's just that's positive. Okay, last call for questions. Anyone else? All right then. Well, we're wishing you the best, Will, and your team to push forward and bring this into fruition. And we can't wait to touch base with you again in a few months and see where you are.
Well, thank you very much, everybody, for your giving me a half hour of your time today. I appreciate it.